everyone, and welcome to Immuno-Tea Time.
Yes.
I am very happy to be here today because today we have our first woman in the podcast. So we are very happy, right, Ben?
First virologist.
First virologist too. So that's so nice. And I brought also my last Spanish tea that I have here in Germany. It's a mixture between green and black tea with some hibiscus and rose petals, so very flowery.
Today on Immuno-Tea Time, we're thrilled to welcome Dr. Andrea Thoma-Kreß. She leads the retroviral pathogenesis group focusing on HTLV1, a human retrovirus linked to adult T-cell leukemia and chronic inflammatory disease. Her research explores how HTLV1 spreads and how we can stop it. One of her key projects looks at how the virus is transmitted from mother to child through breast milk, aiming to understand the underlying mechanisms and find ways to reduce this risk, especially in endemic areas where breastfeeding is vital. As one of the few HTLV1 experts in Germany, Andrea leads one of the most active research groups in Europe. Andrea, thanks for joining us. Let's sip into science.
Do you want to tell us a bit of how was your path until here? Like from when you started, how did you end up in HTLV?
Yeah, thanks first of all for this kind introduction and for having me. It's a great pleasure for me to be the first woman and the first virologist in your podcast. So to be honest, when I was at school, I was always interested in viruses and in cancer. And when I started to study, this interest was still there. And then I decided, okay, I have to do something which combines these two interests. And when I did my studies and started virology, I learned that there are tumor viruses around. And one of them is HTLV1. And I found it a quite interesting model system, since you can not only learn much about this virus itself, but also about tumor biology, because it's also used as tumor model. And so I very early in my career decided to work on tumor viruses and came to HTLV1. And my career path was rather not the normal one, because I did my PhD on HTLV. And while I was doing this, my mentor, he suffered from a cancer and was dying while I was doing my PhD, passed away. And this was very, very sad for the whole team and the whole institute. And our group more or less split. And I was the last PhD student, how to say, but I could continue my work on HTLV1 and finalize my thesis. And afterwards, I got the opportunity to start right now a junior research group. But I did something completely different from that time point, because during my PhD, I was more working on signaling and oncogenic signaling, but I was always fascinated by cell-cell communication and virus transmission. And there's not so much known about virus transmission of this virus. And therefore, I started to do this and got the opportunity for nice funding by the DFG and could start projects focusing really on cell-to-cell transmission of this virus.
Will you say that your mentor helped you a lot to kind of focus on HTLV1?
Yes, because the former director of our institute had a great interest that someone continued this research area here in Germany, because when you look, there are not many people working on HTLV. And well, I was more or less one of these few. And now I have the biggest group in Germany focusing on this virus. And that's what I say. I'm very grateful to my former mentor, but also to the previous one, to the previous director, that I could continue to work on this virus, because there's an urgent need to find therapeutics against this virus. It's really underrepresented in, let's say, public health. And therefore, it's very important that you really understand how is it transmitting, how does this work, and to learn more about molecular details, because just when we know how everything is working, we can directly interfere with it. If we don't know, it's very difficult.
Yeah, I think you have also organized recently an HTLV conference. How was it?
That was really an exciting thing for me to do. Well, we had the European Congress here. We had more than 70 people. And the funny thing was, they were not only from Europe, they were from all over the world. We had participants from Brazil, from Australia, from Japan. So it was really international. And it was a lot of work. But in the end, it was really great to have all these people here. And I had great support from the whole team to do this. And it was exciting because you had 3 days only focusing on HTLV. So every talk was interesting and it was the conference for us. Yeah, so really great. We had nice weather and people enjoyed to be here. And here within the institute and the botanical garden, it's also a nice scenery to have a congress. So that was really success. And they already asked me whether I will host the World Congress. And I said, well, now I'm done for the next few years. Ask me in a few years. But first of all, we need a little bit recovery.
And now talking about a bit of gender in science, because as we said, you are the first woman that we are interviewing in the podcast. How was entering in this field that we almost only hear male names?
Well, I have to say, this differs during your career, because when you look in your master's classes, for instance, you see that gender is balanced. Yeah. Later on, when you look for PhD students, you also see a balance. But then the further you come, habilitation, for instance, it drops down. I have a chart here in my office about researchers, female researchers in Germany, and this reflects quite nicely the numbers. And when it then comes to full professorship, you really, let's say, get to a less frequent number of female scientists. Within my specific field, it's not like that. I have to say, within the HTLV field, we have a balanced ratio of project leaders, but maybe this is due to the fact since it's a neglected virus. I don't know. This could be the case. I would rather say we have more female project leaders than male project leaders. But in science in general, it's still, I have to say, a problem. That's why I'm also actively participating in InfectNet. InfectNet is an association of female scientists working in infection research. And this was established by the University of Aachen under the leadership of Professor Gabriele Pradl. She got some funding from the Federal Republic of Research and Education. And they could establish this kind of network, which is quite nice because with this network, which is now also professionalized, we have the chance to really connect female infection researchers across different disciplines, from virology to microbiology to epidemiology and immunology, and also clinicians. And that's very helpful to also see, okay, we are many, more than I expected, and that we also can network and can connect with each other.
I think that's very cool also to have that message here in the podcast, because we are a lot of young female scientists trying to appear one day in the world. So it's also very nice.
Do you think things have changed since you started in research?
Yes. Well, I have to say, for me, it was never a disadvantage to be a female researcher. Especially when you're a junior researcher, there are many grants where you can apply, which are, let's say, limited to people, let's say, after several years after their PhD. What's also very good is in Germany, with the German Research Foundation, you also can add some breaks in your career in the CV. So there's a certain field where you can add things like that. Okay, I had 2 kids. And they consider this in your application. So this is something that is really good in Germany, that we have this system. And I have to say, for me, it was never a disadvantage. However, what I noticed is the older you get, and since now I also have the habilitation, I have no longer this junior bonus, how to say. So now I'm really, let's say, in the world of the big ones. And there I see that competition is much bigger, and in the end we will see what comes next, but I'm quite confident, and I'm also very happy to see that more female researchers decide to take this path and also to stay in academia, because I have to say, if you have kids and if you really have your own group already, it's something which I would say you can perfectly combine it with family, as long as you have personal, as long as you have people that work in the lab. At earlier stages, when you really have to do the experiments on your own, or when you have to do infection research, it may sometimes be difficult, especially during pregnancy or during breastfeeding, because with some pathogens you're not allowed to work under category 3 or things like that. And there I would say, if you really need a good mentor that paves you the way, that you can continue your research, or that maybe a technician takes over this part for you, and that you can more focus on writing and things like that. But if you have personal and people that do the work for you, I would say you can perfectly combine it with having a family.
Interesting.
I think it's easier than in industry because you have your own group and you decide when you have, let's say, your appointments with your members of the team. Of course, I cannot change the meetings for the lectures and things like that, but it's very flexible. At least if you have, let's say, a nice network, on the one hand, a team, on the other hand, also a nice institute with a very supportive director that gives you the freedom to do it according to your family needs also. And of course, also at home, you always need a plan B, of course, but that's independent of what you're doing. If you work in industry, it's the same, I would say.
Yeah, I mean, if your kid is sick and you need to go, you need to go
always-
if you're working in academia or in industry. At the end, it's the same. So,
But
and now,
the big difference is what you will never have in industry. Maybe you will have more money, for sure you will have more money, but you never have this freedom that you really say, okay, I have an interesting idea. I try to get some preliminary data on this idea. I try to convince somebody who gives me some money for this idea. And then when you have this grant and you hire the person who does this then in the lab, that's great. And the best is like today, because today we have a defense of one of my PhD students, when then some years later, a person worked on this idea and defends his thesis. That's the best day for a scientist because you can say, yes.
I did it.
No, not I, but the team did it, or he did it, or she did it.
Yeah.
But in the end, this satisfaction, this is something that's really amazing. And I'm very grateful that we have this freedom of scientific thinking here in Germany and still have it, because we learned that it can change very fast from some other countries. And this is really good that we still have this, and it's a privilege, I have to say. And that's something I really love about this job.
Will you say that that's your favorite feeling when you're working in this field?
Not only this, I also like to see people develop. So these are two things. On the one hand, to find out something new and to really have an idea and to see then at the end, okay, the idea was right. It's also interesting if it wasn't right, because you always learn. Yeah, you always learn by doing the experiments. And sometimes you think it's so easy and in the end it's so complicated. But if you never do the experiment, you never know. And this is one part, to find out something new that no one ever has done before. That's very unique. And the other thing is to see people develop. When you really have students in the lab, when they do their course, and then they come for some student assistant jobs, they do their thesis, and then they go and continue and do their PhD. And when you then in the end really see how they develop, and that's great to see. And when they then go and you see them years later and they did a postdoc somewhere else, and you really see, okay, you could really guide them a little bit, but then you see how they start to fly.
Okay, so now if we get into topic, we are going to talk about HTLV, that we know that is a human retrovirus that infects T cells. And so what does retrovirus mean?
Well, retrovirus means it's an RNA virus that infects a cell. And then this RNA is reversely transcribed. And that's where the name comes from this reverse transcription process. And this DNA that it exists is then integrated into the host cell. And this means that the retrovirus, let's say, changes also the genome of the target cell. It integrates, and this is required for producing new viruses. So in the end, when new viruses are produced, other cells are getting infected. But what you always have this retrovirus with you, so you will never get rid of it. And that's the problem we have, especially with this HTLV, because this virus carries oncoproteins, and by this you integrate an oncoprotein into a cell. And the retroviruses have been studied in different organisms, and there are many oncogenic retroviruses in other species, for instance, in mice or in birds. But in humans, HTLV, the human T-cell leukemia virus type 1, which was discovered at the beginning of the 80s, is the only human oncogenic retrovirus. and it's related to HIV, the cause of AIDS, but it's different. It's a different family. So HTLV is a delta retrovirus, while HIV is a lentivirus, and they also differ in the composition of the genome. They are both complex retroviruses, so they not only encode for GAC, pol, and envelope, which is, let's say, the standard retrovirus, which makes you the virus itself and encodes, let's say, for some enzymes that are required for the production and the replication of this virus, and which encodes you an envelope protein. But they also have some additional proteins, regulatory protein and accessory proteins, which makes them complex retroviruses, because the simple ones have just this GAC, pol, and envelope, whereas the others have additional regulatory and additional accessory proteins. So it's a complex retrovirus.
So why is it complex? What's the benefit of for retrovirus to be complex?
Well, the benefit is by having these additional proteins. On the one hand, they can foster their own transcription, because those regulatory proteins encode a transactivator protein. For instance, for HIV, it's the TAT. For HTLV, it's called TAX. And what they are doing is they foster the transcription of this virus. And they have this kind of boosts virus transcription. And this is an advantage compared to retroviruses that lack these proteins. On the other hand, they have nice accessory proteins that really boost viral persistence. For instance, HTLV encodes a protein that is mobile and enters target cells, and it's supposed to dampen immune responses in these target cells, and therefore is important for establishing virus spread and virus persistence. And it's always beneficial to have some additional helping proteins that favor virus persistence or that interfere with immune response. And that's exactly what's going on with these additional proteins, that they are beneficial for virus replication, but also beneficial for maintaining the virus within the body and to establish the infection.
But for us, these extra proteins are involved in causing cancer and diseases, right?
It's for HTLV, it's like that. We have this transactivator, which is on the one hand enhancing virus transcription, but it's also an oncoprotein. So it deregulates a lot of cellular signaling pathways. But also retroviruses that don't have these oncoproteins can cause cancer, because it depends where they integrate into the host cell genome. But the likelihood is much less than when you have a virus that brings its own oncoprotein. So, and for HTLV, it has an oncoprotein, and it was found out very early that this oncoprotein is sufficient to transform primary human T cells to permanent growth, on the one hand, that it can cause tumors in immunocompromised mice. That's always an indication, okay, there's something oncogenic, and that it also can really deregulate a lot of signaling pathways that are associated with cancer. And the bad thing about HTLV, it has not only one, the transactivator, it has a very special protein and RNA encoded in antisense. That's quite peculiar for this virus. And this is also known to really to interfere with oncogenic signaling and to foster the maintenance of this oncogenic phenotype. So it has not only one, but two of kind of oncoproteins, which makes it very oncogenic. And there was a comparison. It was a paper from Bob Gallo where they compared the oncogenicity of different carcinogens. And when you really consider this, and it's quite crazy, because if somebody is infected with HTLV, the lifetime risk to develop a leukemia is round about 5%. But this is only due to the fact that patients usually develop the leukemia very late in life. It's an adult T-cell leukemia because patients are adults when they got the leukemia. However, there were some calculations. If you really get the virus early in life, for instance, via breastfeeding from mother to child, the lifetime risk increases up to 25% to get this cancer. And that's ridiculous, because you could smoke 20 cigarettes a day and your risk would not be that high to develop a cancer. And this is my comparison when I tell somebody about this virus, to tell them it's very oncogenic. due to the fact that it has 2 oncoproteins.
So a person that can get infected with HTLV, so like mainly it's asymptomatic, right? Like the disease, but it can be that you end up having leukemia.
Yes.
Why?
Yeah, that's the big question. And this is something where really many people work on to really get to know why do some people develop a cancer and others don't. Because we know, as you said, many stay asymptomatic. To be honest, this asymptomatic is always written in the textbooks, but there were several studies that found out that these people are suffering from different issues. They have some health problems. And in the field, people now tend to say they are people living with HTLV rather than asymptomatic, just to say, okay, they have this virus and they always have the fear and the risk to develop the disease. What we know is that a risk factor to develop a disease is the viral burden. So what we can measure is how many copies of the virus do we have integrated in the cells. And then we can measure the so-called proviral load. It's a PCR-based diagnostics tool that tells us how much virus per cell. And the higher the proviral load, the higher is the risk to really develop the disease. So by this, we have, when we monitor patients and they have a low proviral load, one would say, okay, their likelihood to really develop the disease is low. But this requires that you really monitor patients. And as we heard at the very beginning, this virus is endemic in countries where monitoring is, well, not frequently performed. For instance, we have a large unknown risk of HTLV in Africa. We have South America, where we have endemic hotspots. We have central parts of Australia among Aborigines. We also have industrialized countries like Japan, where we have a high frequency of HTLV. There, the monitoring is great. But to be honest, in developing countries, it's very, very difficult because people sometimes even don't know that they're infected. So we have one measure, this is the proviral load. But what we also know that certain polymorphisms or genetic associations are associated with an increased risk to develop diseases. And there are several studies ongoing, genome-wide association studies that really look into more detail into this. What I can tell you, it depends on where you're looking. So results, for instance, from Brazilian cohorts are different from those of Japanese cohorts, for instance. So there seem to be geographic differences and genetic differences. What's also known is that if you develop this leukemia, you can already predict this several years in advance because the virus is integrating and some of these clones, this is what they are called, are growing out. And by measuring these clones and the distribution of clones, you can get a kind of prediction whether a person has a higher likelihood to develop the disease or not. This is extensively done in UK, in London. It's Imperial College, where some research groups are really addressing these questions since they look in patients before they develop the leukemia and at the stage when they started to develop the leukemia, and to really backtrace what can I use as a kind of predictor to really say, okay, we should have a closer look at this patient and should maybe start already with an intervention.
And what can you do when you find out you have HTLV?
Well, when you have HTLV and you're asymptomatic or don't have any symptoms, usually you don't do much. It's because, for instance, it depends on also where you get this diagnosis. If you get it, for instance, in London, you're a lucky one because there is an HTLV1 clinics. Or if you get in Sao Paulo, you're also, let's say, lucky one because there are experts, MDs that work on this virus. If you get this diagnosis somewhere else, in Japan, you're also lucky one. But for instance, in Germany, you're lucky one that you're in Germany and that you can do all these diagnostics. But even in Germany, the doctors, they heard about it, but they don't know much about it. And there's really a gap of knowledge. And why? Because they don't have many patients. So it's not like, okay, I do the treatment as I do it always. maybe they only have one HTLV infected patient during their career, and that's something new for them. So they first have to find out, what shall we do? So if there is somebody who has HTLV but has no symptoms, what you would usually do is to say, okay, we measure the proviral load, let's say once or twice a year, and send this person to a neurologist and hematologist just for having a look and for controlling the patient. However, as soon as you have some symptoms, like neurological symptoms for the HAM-TSP, the best thing is really to get enrolled into a clinical trial, because the standard treatment differs also where you're looking for, and it's mostly symptomatic for HAM-TSP patients, so that they don't suffer from so much pain, because HAM-TSP is very, very painful. Patients suffer from walking disturbances and lower back pain, so they need a wheelchair over time. They cannot walk any longer, and it seems to be very, very painful, according to what patients tell us. So usually they get symptomatic therapy and physiotherapy, something like that. For ATL patients, usually when they get the diagnosis, it depends on which subtype of ATL it is, because there are more aggressive ones and there are chronic ones. And the chronic ones have a better prognosis than the aggressive ones. For the aggressive ones, the lifetime estimations range from 4 to 8 months. That's not much. What people try to do is to get these people, and what doctors try to do, to get them into clinical trials. And this is usually chemotherapeutics, what they are doing with them. In Japan, they also now use a monoclonal antibody, and what they try to do is that they get a stem cell transplantation, because this is, like for other leukemias as well, a possibility to cure. The problem is with the stem cell transplantation that patients that suffer from ATL are usually very old, and the older you are, the worse is the likelihood that you really have a good outcome after such a stem cell transplantation. And it depends also on the subtype of ATL that you have, and on the pre-treatment, whether you then have a relapse or not. So there are many studies ongoing in Japan where they use monoclonal antibodies against CCR4. It's a marker that's upregulated on these ATLL cells. And this seemed to be quite successful. But now we know that it also depends on when you give this antibody, for how long you give this antibody, and what's the distance between giving this antibody and starting the transplantation. So there is still a lot to be learned to improve the outcome of these patients. But usually it's a chemotherapy that they get.
So when someone has HTLV and they develop disease, you can have a leukemia, right? And then a neurological disease. Is that right?
And even more. These are the two most common ones. So we know on the one hand that it causes this leukemia, lymphoma, which is also very infiltrative. So they also have metastasis. And on the other hand, as you said, we have this neurological disease, but we could rather say it causes inflammatory conditions. It's not only the neurological one I was talking about. We have a bunch of diseases that's associated with HTLV, but those two are the most frequent ones. So around about 3 to 5% of infected patients develop either the one or the other. So in total, roughly 10% develop a disease. But the other ones are, let's say, not as frequent as the ones you were mentioning before. But they always have to do something with inflammation, so arthritis, uveitis, things like that. And what we also have, especially in certain countries, are co-infections with nematodes, for instance. But this is not globally distributed, but this depends also on the country where you're looking at.
But I imagine it's in a lot of endemic areas like maybe Central Australia or in parts of Africa where the healthcare isn't that good, co-infection is quite common, right? And does that make the disease worse?
Yes, this depends on the co-infection, because for instance, for strongyloides, this is a nematode, there have been several studies that have shown that the diseases or the two pathogens influence each other, and that you have, for instance, you influence the risk to develop disease because you have an interplay between these two pathogens. But to be honest, there are only few studies and they only focus, let's say, on patient cohorts, and these cohorts are usually very small. But what is really lacking are larger studies where you really look on co-infections and really see what are these two pathogens doing with each other. So this is really a big gap of knowledge, this co-infection part, where you really focus also in the lab, that you really do experiments and see what is going to happen. And the data we have is there, but only from very few studies. So it's not, that's the problem, because as I told you, it's a neglected virus. And I'm always asked, we know this from HIV, how is it with HTLV? And then I said, well, I don't know. You don't know? I said, well, not only I don't know, it's not known. Yeah, that's very frequently a problem. Also, when you think about structure of certain proteins, or, we have this and that antibodies for N, for this version of envelope protein and for that version of envelope protein, you could do the same with HTLV. Oh no, we can't. We even don't have the structure of the envelope protein. So this is always what you have to say when people are asking you, that many things we know for other viruses, we don't know for HTLV.
And why do you think that happened? Like, why do you think HTLV didn't have that much
power?
Yeah.
Well, I think it had to do maybe also with how many people are infected worldwide and who is infected. And is it a problem in industrialized countries or not? So we have a strong HTLV research in Japan, but they also have many HTLV infected patients. So Japan has a certain interest to do something about HTLV, and that's very good because there are many, many brilliant scientists and studies in Japan. However, for instance, in other countries where this virus is, let's say, under the radar, there is not that much, let's say, political interest. I'm very, very glad that in Germany I got some funds from the German Ministry of Education and Research to really give us a lot of money to study mother-to-child transmission of HTLV. And my colleagues were quite fascinated to hear that they supported us that much. But I think what they realized is that it's a global health concern, because also the WHO has put HTLV on the list of these candidates of sexually transmitted diseases that shall be eliminated. And there is a political pressure, but this pressure is there due to a lot of advocacy out of the HTLV field, I have to say. And this is something I didn't expect, because I thought, okay, when I work with this virus, I do my research and that's it. But it's not like that. You really have to communicate what this virus is, what it is doing, and that it's a global health threat. And if we find strategies to really interfere with cell-to-cell transmission, that would be the easiest step, because by this we could really stop the virus and don't have to develop expensive therapies, but just by prevention strategies, we could really combat this virus. That would be the easiest way. And of course, vaccination, but there is no licensed vaccine yet. There are some vaccines in preclinical models, how to say, in mouse models or other animal models that are quite promising, but there is no licensed vaccine on the market.
So could you tell us a bit about how HTLV spreads, and then maybe we can go into what milk TV is?
What milk TV is, yes. Okay. So the virus spreads quite different from other retroviruses, because usually when you think about a virus infects a cell and produces new virions, and these viruses then infect other cells. And when you work with viruses, you can isolate viruses and put them on target cells, and they infect these target cells. For HTLV, it's a little bit different, because the cell transmission is completely cell-associated. So that means if you have an infected cell, it needs a close cell-cell contact to a target cell to really infect this target cell. And I'm here talking about T cells, the CD4 positive T lymphocytes, the major target of HTLV1 in vivo. So if I take 3 virions and just put them on target cells, I will never achieve, let's say, a good rate of infection, because the T cells, they need the cell-cell contact. This also explains us how this virus spreads. because it always needs cell-containing body fluids. So this means you have transmission from mother to child while breastfeeding. This is where our Milk TV project steps in. You have transmission via cell-containing blood products, and you have transmission via sexual contacts, or quite rare and not maybe in industrialized countries, but where you have bushmeat and things like that, bushmeat, there you really have transmission via contaminated blood or contaminated bushmeat, because it's also in some monkeys where we have a related virus that can jump to human. Yeah.
So what do you think is the most common way that people get HTLV then? Is it from mother to child?
Mother to child and sexual transmission are the two most common ones, and it depends on the country where you're looking for. So, but those two are the top candidates.
Okay.
And for sexual transmission, all those measures that help to prevent HIV also help to prevent HTLV. And for mother to child transmission, the situation is different, because for HIV, we know that people that are on antiretroviral therapy for HIV, that they can breastfeed their children. However, for HTLV, we don't have any therapy. So there are no studies that ever tested antiretroviral therapy in a clinical setting in mother-to-child transmission. And that's really a problem, because if we have a mother that's HTLV infected, we can just currently recommend not to breastfeed. And that's really a problem if you think about where we have the virus, because when you recommend a mother not to breastfeed, this also implies that she has to take formula. Formula is expensive. So if you tell a mother not to breastfeed, you must somehow provide her the money that she can buy the formula. The second point you need is clean drinking water. And this is also an issue in several countries where HTLV is prevalent, because not everywhere on the globe we have access to clean drinking water. And if you don't have clean drinking water and provide the formula with dirty drinking water, you have the problem that these children may suffer from diarrhea and get some other infections, which may then be even more worse at first, because they can die from these co-infections. And that's always the dilemma where you are, that you on the one hand have a virus that is transmitted from mother to child, and that breast milk is highly recommended for the nutrition of the infant, and also by the WHO for the first 6 months in life. But on the other hand, you have a situation where you cannot easily say, oh, stop breastfeeding. And for instance, in some countries where they really try to start these policies, where they do the screening for HTLV in pregnant women, they have implemented some programs that they also provide the formula for the mothers. For instance, in Brazil, they did a great job during the last years, and this has started just a few years ago, that they really also provide the support then. But there are still so many countries where we don't do the screening at all, and where mothers are left alone and are not advised anything. And what we want to do is that we would like to find a way, first of all, to better understand how the virus is transmitted from mother to child by breast milk, and to find a way to develop strategies that the mothers can still breastfeed and without transmitting the virus. Because by this we would have, let's say, a solution for this dilemma, that we can on the one hand say the infant can really benefit from all these positive aspects of breastfeeding, but the mother and the infant don't have the risk of this mother-to-child transmission. That would be our dream, how to say.
So you will try to load the proviral load and then expect that it's not transmitted anymore.
There are several strategies how you could do something like that. One strategy would, of course, be to test whether antiretroviral therapy also works for HTLV. The problem is, as I told you, the transmission is different. Well, HIV also transmits from cell to cell, but it can infect cell-free as well. And for HTLV, what we have, when it's integrated, you can have an expansion of these cells where the virus is integrated. So you don't necessarily need a de novo infection. And if you really do antiviral therapy, you would always interfere with an enzyme that's required for new infections. But if I have an infected cell and this infected cell can somehow spread in my body, an antiretroviral therapy will not be beneficial. But if an infant gets infected, we expect that at least at the beginning of infection, we have new infections. Why do we expect this? Because we see that there is a distribution. We have different integration sites, so there must be some de novo infection. And it's not just one clone that we say it's one cell that was transmitted, but we see that the virus is integrated at different times. And this argues in favor of we had a really active replication. So it's quite likely that we could also somehow interfere when we use antiretroviral therapy. And a colleague from in UK, Dr. Martens, she has really beautiful in vitro data that shows that integrase inhibitors are effective against HTLV in vitro, and these integrase inhibitors are on the market for HIV. So there's really a strong need to retest now these integrase inhibitors in context of a clinical setting, because they are already on the market, but no one has ever tested them for HTLV1 in a clinical setting. So this is one big need we have. But my research is more centered on different strategies. We try on the one hand to find a way to understand the interplay between the milk and the virus, and also whether we can find some risk factors or some factors in the milk that favor virus transmission or don't. Because what we know, the risk from a mother to transmit the virus to her infant increases with increased proviral load. So the more virus the mother has, the higher is the risk that she really transmit the virus. On the other hand, what we also know is it depends on whether the mother has ever transmitted the virus to one of her offspring. This also increases the risk. We know that genetic factors may play a role, like HLAs. We also know that the income plays a role. But what we didn't know so far, and what we try to uncover, is how the milk itself somehow provides the environment that the virus is transmitted or not. And this is something we are studying, because we are really looking on cohorts of mothers where we look how the milk is influencing virus transmission and whether certain components of breast milk impair virus transmission. And we really have nice results that are currently, we rewrite this, it's currently under submission, where we really could find that some mothers have some components in milk that seem to act antiviral, while others do not. And the goal now for the prolongation of our project is to really find out what's the difference between these mothers. Because if we really see and find something in milk that could predict whether it's likely that a mother transmit or doesn't transmit, this would be really interesting, because then we would just need a drop of milk and can give you a kind of indication of, okay, you are combined with other things as well. Your risk to transmit this virus is high or is low. Because in the end, we would like to keep the mother breastfeeding. And this is something that's the difference from others that say, okay, let's stop breastfeeding, because we feel that breastfeeding, especially in these resource-limiting settings, is the best you could ever do by avoiding all these complications with diarrhea and so on. On the other hand, we are also interested in the envelope protein of the virus, because, as I told you, the virus is like a normal retrovirus, so it encodes this envelope protein, and this is required for entering target cells. And what we try to do is to block the binding of this envelope protein to target cells, and there we are working with small molecules called nanobodies. They are single domain antibodies and usually produced in llamas. And they are very small compared to conventional antibodies. And this may be an advantage, because they may recognize epitopes that are not recognized by antibodies. And the big advantage of these nanobodies is that they are also resistant towards acidification. So they are quite stable. And there we already have now some of these nanobodies. And we are currently developing and further developing these nanobodies towards specific ones that, and our goal is to really have ones that really block transmission. But this is not so easy, because when you immunize and isolate nanobodies, you never know what you get, and you first have to test all of these different nanobodies. And of course, the outcome depends on with what you immunize, with what you do your panning to really get binders. So this is something where we still are, let's say, work in progress.
So is the vision to have these nanobodies which you then take as a medicine, or you put them into milk which you can then give to a baby? Or
different strategies? Of course, the best thing would be to have the nanobody already there where the virus enters the cell. And there's the next problem. No one knows where this is the case. Because for HIV, we know tonsils play a role. But for the oral route of mother-to-child transmission, this is enigmatic. We really do not know. We have a nice collaboration with a team from France where we have the possibility to study different organs in an in vivo animal model of HTLV1, of a related virus, and where we can look in different organs. And this is very helpful for us, because what I can tell you, but I can't tell you more since it's not published yet, the tonsils don't seem to be the ones that are the ones with the highest viral load. So there seems to be a difference between HTLV and other retroviruses. And when we really know where the virus enters, we know where we have to redirect our nanobodies to. First of all, when we started this project, I was thinking about something like, okay, maybe we can use a kind of application which the mother can take, and when the infant is suckling, that you just have the nanobody in the mouth of the infant. But this is something which is really different to get realized in a setting where you have let's say resource limited settings. So in the end, it must be something like an immunization or something like that, that you really have a specific immune protection directly at the site where the virus enters the cell. And an alternative we were thinking about is whether we could manage to package these nanobodies into extracellular vesicles or something like that. But then we need, of course, a durable representation of these nanobodies. So there we are really at a very, very early step. So in the end, I'm very sure that the combination of different strategies will really help to find a solution to do so. And also in combination with, because I'm not so, well, to be honest, I'm not sure whether just an antiretroviral therapy would really prevent the risk to transmit the virus, and whether this really works in vivo. So I think it's always important to have an open mind for alternative strategies and unconventional strategies as well.
I mean, right now there's no way you can sterilize the breast milk, or if you have HTLV, the only choice is not breastfeeding at all?
Well, what you could do, you could pump the milk, and there has been some success with some freezing protocols, but these studies are very, very old. So they were freezing the milk and then feeding the milk. But this is something you could also not easily do in resource limiting settings. And for sterilizing, the problem is that you really also get rid of some components that are important for nutrition. In milk banks, they do this so-called holder pasteurization, which is a short-term treatment and heat treatment of the milk, and which seems to be quite okay. But in the end, you have to do this with every milk you generate. And this is something I can tell you because I have 2 kids. I'm not so sure whether this is something you could really do, and whether this is something that could be practically done. So what we would need is really a kind of 1-shot treatment, or let's say a 2-shot treatment or something like that. But this where you really have to treat every milk sample that you feed. That's rather difficult.
That's a lot.
Yes, and I also have to say these studies were just few studies where they froze the milk, and still we don't know what's the optimal freezing protocol and why it's doing that, and why we see some beneficial effects. And this is also something we want to find out, because what I could also envision, if we really find out which components in breast milk affect transmission, maybe it's also possible to really change the composition of breast milk in the mother, for instance, by what they eat. Fatty acids, one example. There are some supplements that breastfeeding mothers are taking, like omega-3 fatty acids and things like that. And this has not been studied at all, how this affects breast milk composition, we know, yes, but how this affects virus transmission. And I'm rather like thinking out of the box to really, to explore when we change something a little bit, maybe in the nutrition of the mother, could this somehow also impact virus transmission?
Yeah, I also wanted to ask you, because we talked that there is a possibility of having antiretroviral through the milk. So maybe that's going to be from what the mother ate, or in comparison with other moms or something like that.
Mm-hmm. Yes, so if the milk itself, you mean, is somehow antiretroviral, yes, this could exactly be, because the funny thing is, and that's also good that we have this situation, not every mother that is infected is transmitting the virus.
Exactly.
And it also depends on how long they breastfeed. This is something I didn't mention earlier. So the longer they breastfeed, the higher is the risk that they transmit the virus. But in the end, nobody knows exactly why. Of course, it could be that the decline of antibodies plays a role, yeah. But there are so few studies that really analyze this in more detail. And I'm very grateful that we now have the unique opportunity with the continuation of this Milk TV project to look in a cohort of infected mothers with a Brazilian collaboration partner at the University of Sao Paulo. They frequently see HTLV infected mothers, and we will collect the colostrum from these mothers and compare this with the colostrum of uninfected mothers. And we will look for everything we can do because on the one hand, we are very much interested, of course, in cytokines and the cytokine levels. And we are also looking by metabolomics and lipidomics and look on the milk composition itself and on milk content and like human milk oligosaccharides and things like that. And to really have a global omics analysis of these uninfected and infected mothers. And this gives us really a good opportunity and a unique chance to learn more about also whether breast milk differs between infected and uninfected mothers, or whether we see some factors deregulated or not. And we also have a questionnaire, and there we are very much interested in whether the mother has transmitted the virus earlier or not, because usually they don't have just one kid, but several kids. And what I didn't know, and which is quite helpful for our study, that there are many elective cesarean sections in Brazil. My colleagues told me that more than 80% have elective cesarean sections. So what we can learn out of this is that if the virus is transmitted from these mothers to their kids, then it's very likely that it's via breastfeeding, because by cesarean section, you also reduce the very, very, very low risk of in utero transmission. And why do we study colostrum? Because in Brazil, there is a general recommendation that mothers stop breastfeeding and that they use supplements and formula milk. And so we can only study colostrum, but not at a later time point, because then this would also be unethical if you really don't have a solution for these mothers and then advise them to continue breastfeeding, although you know they carry a virus that's transmitted from mother to child. So this is very difficult.
Yeah. Now, so we are talking a lot about preventing the transmission of the virus. Would it be like an option of cutting out the virus from the genome? Because we have talked that the virus is getting into the genome of the T cells. Can we just cut it out?
Yes, that's a brilliant idea. And we contributed to the study that was published 2 years ago in collaboration with Frank Buchholz from the Technical University in Dresden. And there are two ways that are, let's say, well known how to cut out some DNA. And one would be CRISPR-Cas9. So you all heard about this CRISPR-Cas9 technology. And the other one would be recombinases. And recombinases are enzymes produced in bacteria. But usually they don't recognize a retrovirus. But what our colleague Frank Buchholz did, he can evolve recombinases that they can recognize a new target sequence, and he can do this in the lab, and it's quite cool. It's a kind of evolution in the lab, a molecular evolution in the lab, where you use a bacterial recombinase which has a certain target sequence, and then you modify this target sequence and modify it according to the LTRs, the long terminal repeats that are flanking the HTLV1 genome. And this is what they exactly did. They used their initial sequence and modified this over time. And in the end, they had the target sequence that is flanking HTLV1. And by doing so, and I think it were around about 180 cycles of molecular evolution, they could generate a recombinase that then recognizes the LTRs of HTLV1. And we contributed to the study because we did all the infectious work and used this recombinase to see whether we can really excise the virus from a piece of DNA that encodes the whole virus as kind of a molecular clone, where we can really see, okay, it excises the virus that it's flanked by the LTRs, and whether we can prevent de novo infection by having this recombinase presence. And what I can tell you, in proof of principle, it works. So you can really use these enzymes and can cut the virus. But what I can't tell you whether this works in every clinical setting, because HTLV is known to undergo several modifications in its chromatin. So we know that it has some deletions, especially also in the long terminal repeats, and that it has encodes for intragenic silencers and enhancers, which may change the architecture of this virus when it's integrated into the DNA. And this has not been studied yet, whether these recombinases work in every clinical setting. But we had this proof of concept study that this is possible. For CRISPR, I can tell you that some people, there are some papers where they really try to CRISPR out the virus, and this has been shown that it works. But the problem for the CRISPR was that you also had sometimes off-target effects. For the recombinase, the good thing is you don't rely on the cellular repair machinery, because the recombinase does the job completely independent of any cellular repair enzymes. But for CRISPR, you rely on the cellular repair machinery, and this can cause errors. And on the other hand, the specificity of the recombinase is really great. So we had no off-target effects. But for the CRISPR, we also tried it, but we never published it. It was just a kind of experimental control for us. What you have is, when you design these guide RNAs that recognize your target sequence, it's very difficult to really have a guide RNA that's so specific for the virus that you don't have any off-target effects. But, also this technique has shown to really get more and more specific, and there are already some CRISPR therapeutics in patients. So I'm very confident that this will be also a good strategy if we further develop these methods, and I would go for both of them, to really find a solution to get rid of the integrated virus. But as I told you, there is still a lot of work. There is no single work done, for instance, for the recombinase in an animal model of HTLV1 infection. It was just done in cell culture and cell lines. It has never been done in any fresh patient material isolated ex vivo. And there's still a lot of work to be done.
So in an infection with HTLV, the virus inserts its code into the genome.
Yes.
And has these LTRs, which are the signal to produce more virus.
Yes, there's the promoter in the LTR. And this provides a signal to produce more virus. But the funny thing is for HTLV that the virus gets latent. So when you really have a patient, usually the sense strand of transcription is silenced, and you have the virus, let's say, in a kind of a latent version. And this is a kind of immune escape mechanism, because the virus encodes a very immunodominant protein, and we have a strong CTL response targeting this immunodominant protein. And by keeping the virus silent, you evade the CTL response, and this keeps, let's say, the virus alive, because it evades all these detrimental immune responses.
So, and then with the recombinase, you can just put this protein into the cell, and then just cut the virus out, and then that virus is destroyed, and then the cells aren't infected anymore at all. Is that right?
Yeah, that would be the concept, but in the end you would have still a little bit of a piece of DNA from the virus, but this shouldn't be harmful. Of course, it depends where it has integrated and what happens when you recombine. because you would, of course, lose all these viral proteins and all these oncoproteins, what I told you earlier, but you still would have a little bit of DNA, a piece of DNA left from the LTRs, but this shouldn't be harmful. But still, it's somehow a manipulation of our DNA, and this is always, let's say, an issue, because if you have something that integrates somewhere in your genome, you cannot say it's without a risk. Yeah.
Very interesting. So what do you think if this therapy with the recombinants becomes like mainstream and in years we can have it, do you think we will get rid of this virus and it will never exist again?
Well, what we need, of course, even if we have the perfect therapy, what we need is awareness, global awareness, that people are screened, that we know who is infected, because we can only get rid of the virus if we really find the patients. But if we still have this high frequency of people that even don't know that they have the virus, how can we treat them? And one big issue, and where we really have to combine forces also among the researchers working on this virus, that we really foster programs on screening of this virus. I can just tell you a story. In the UK, they have been trying, I think for more than 10 years, to put HTLV1 on the list of antenatal screening for the National Health Service. But it has never been put on this list, and I'm European representative of the International Retrovirus Association. That's the professional society for people working on HTLV1. And they were asking me whether I can also write a letter to support this need for antenatal screening in the UK. And there were several other people that did the same. And in the end, we didn't succeed. But what we now have, at least, that they said, okay, we will screen risk groups. So risk groups means people from HTLV endemic regions. And at least this is a partial success, because we know that in certain populations the virus is more prevalent than in others. But what we also know, due to migration within the last years, due to wars globally occurring, we have a change in the populations. Also in Germany, when you remember the last 10 years here in Germany, the population has changed. We had a lot of migration. And we have to consider also this when we think about infection prevention, that a population changes over time. And and we can only be successful when we really provide screening.
So I think that now, as we are reaching the bottom of our cups, it's time for the last sip question. That is 3 questions that we ask all of our guests.
Okay.
They are a bit of philosophical, so you can take your time to answer. So the first one is, if you could go back and speak to yourself on your first day in the lab, what would you say?
What I would do different, or what do you think?
What would you say to yourself? Like, would you have any different advice? Like, okay, maybe don't do this anymore, or follow your thoughts. I don't know. Just as an advice also for us that we are starting now.
Well, I think I wouldn't change that much. What I would say, always stay optimistic. And that's what I also tell my students and the doctoral researchers. Never lose the trust in your idea, and stay optimistic. And also, if the experiment fails, you will learn something out of it. And even if it's a negative result, if it's technically sound, it will help you in the end. But you have to find this out. And this is something that's sometimes a little bit tricky, especially for the students. But never lose the trust in your research.
Yeah. Yep. Learn how to deal with frustration.
Yes, that's very important. That's very important. But you have to really, this is something you can learn.
Yeah, also very important in life, not only in science.
Yes. But if you work as a scientist, you will be trained quite hard in your professional career. This may also help you for your private life, of course.
Okay, the second question is, what's one mystery, scientific or personal, that you hope to see solved in your lifetime?
Of course, what I would love to solve is to find a strategy to prevent this mother-to-child transmission and allow the mothers to still continue breastfeeding. If I can contribute with our research to this, I think then we would have achieved really a lot, because on the one hand, we have the babies that get their proper nutrition, and on the other hand, we have the mothers that don't have the fear to transmit the virus. So this would be my lifetime goal, to really help with our research, that we can help all these mothers and their babies.
Yeah, I think it's great. It's research with a real tangible way to help people.
Yes.
It's really important.
And really something that reaches the patient and the people living with this virus, because sometimes you do some experiments in the lab, transfect two plasmids, and okay, they're interacting or they are not. But in the end, you always have to ask what's going on later on with this stuff. So what will be the benefit for the patient? And this is something I also had to learn, because I started with exactly this, transfecting two plasmids and looking what they are doing. And we still do this because it's important, because if you don't know really how it's working, and you need to know this, you need to go into the molecular details. But what you always need to know, and what you never should forget, is what is the benefit in the end to combat this disease, or to the benefit for the patient.
Yeah. And our last question is, if you could ask the immune system one question and get a clear, honest answer, what would you ask?
Why, dear immune system, are you not able to eliminate this virus upon de novo infection?
It's a good question.
Yeah. Let's see what the immune system answers.
Well, maybe I dream about it tonight. I don't know.
Yeah. So thank you very much. I think it was a very nice conversation.
Yep.
We learned a lot.
Yes.
So.
Take-home message: HTLV is a really important virus.
Now everybody knows about it. Everybody researches about it, and
listeners, go look up HTLV and consider getting screened for it if you're interested in having a child. Right.
Well, it depends, of course, where you get the child. So the risk in Germany is very low, to be honest. But it's very important that we raise public awareness about this virus. And also thank you for your support, because by doing something like this also helps to raise awareness.
Yeah.
Okay. So maybe then the message of the podcast is go out and tell a friend or family member about HTLV and try and let someone else know about it.
Yeah. So stay curious.
Stay critical.
And see you next time
For some tea. Great.
Thank you very much.
Yeah, thank you.
Reach us on Instagram at iimmunefau, or send us questions, ideas, or feedback to iimmunefau@gmail.com. The equipment for this podcast is graciously provided by the Friedrich Alexander University Rundfunk Studio and Thomas Bauernschmidt.